Hello, all. Welcome back. We will cover our first organism in our organismal diversity unit. This is actually a domain of organisms called the prokaryotes. We have two domains within our prokaryote distinction, domain bacteria and domain archaea. Let's see what makes a prokaryote and learn some interesting things about them. Prokaryotes are unicellular, so only one cell, very tiny, organisms without a nucleus and no other membrane-bound organelles. We've actually learned this before, way back in chapter 4 when we were learning about types of cells. We learned about prokaryote cells and eukaryote cells. These are the prokaryote friends, and the word itself can give you the definition. So pro, Latin for before, and karya, Greek for kernel, or at this point we know it now as the nucleus. So these organisms were thought to have been created or evolved before those with a nucleus, so prokaryotes. They are ubiquitous. They are found everywhere that there is life, and also places there is no other seeming life that we can see with the eye, including on and in our bodies and bodies of other multicellular organisms like plants, other animals, fungi, etc. And they outnumber eukaryote cells 10 to 1. Here is the basic structure of a prokaryote. So we can see, and again, this is something we've already learned back in chapter 4, so we're revisiting this now. They have no nucleus, so again, their DNA just exists in this free-floating clump that we call a nucleoid. Similar word to nucleus, but it's a nucleoid. Of course, they have cytoplasm, ribosomes, and a plasma membrane, so those four things were the universal features of all cells. But in addition to those things, they also have a cell wall. Usually we'll have some sort of capsule, which is like a sticky coating that can either secrete poisons or help with adhesion. And then various movement appendages, either tiny little hair-like structures called pili or a long whip-like tail called a flagellum. When trying to identify and define what kind of prokaryote we're dealing with, oftentimes we can use the shape of the cell and how they are arranged. So for example, if we have a singular cell, just one by itself, there is no arrangement. We would just determine what is the shape of that cell. So a single bacteria cell that is in a rod form or an elongated form, we call a bacillus, or a bacilli is plural. If we have a singular bacteria that is sphere-shaped, we call that a coccus or a cocci or cocci if it is plural. So definitely be able to distinguish the two types of shapes. And by the way, there are many actual shapes for bacteria cells, but we will just learn these two. And if we get more complicated, we get into arrangement. So if we have two cells connected, that is a diplo arrangement. So if we have two spherical cells, that is called a diplococci for two circular cells. If we have two bacillus cells, we call that a diplobacilli for two bacillus cells. Then we get into more complicated arrangements. We have a strep arrangement, which is long chains. So for example, if we have long chains of bacilli, that is a streptobacilli. And we also have clusters, a staff arrangement. So if we have clusters of cocci cells, we can now call that a staphylococci. And staffy or staff is actually the Latin prefix for grapes, like a bunch of grapes. And these prefixes for these more complicated arrangements may sound familiar to you. So, for example, if you have strep throat, you actually are infected with a streptococci bacteria. So, long chains of little balls, just like this. The doctor will swab your throat, inoculate it, look at it under a microscope, and if he sees long chains of circles, then he would probably go ahead and diagnose you as having strep throat. Staph is also a pretty common disease. If you've ever had a staph infection, then you are infected with a Staphylococcus bacteria. And a lot of those are very notorious for being hard to get rid of. If you have ever heard of MRSA, which is a type of staph infection, MRSA stands for methicillin resistant Staphylococcus aureus. So that type of staphylococci, that specific staphylococci, Staph aureus, is resistant to a very common antibiotic called methicillin, which is why staph infections can be very hard to clear up. So you go ahead and try to name this bacteria, and on an upcoming quiz or exam, I will show you various shapes and arrangements, and I want you to be able to name them. So what would the name of this guy be? This would be a staphylococci, so a cluster or a bunch of sphere-shaped bacteria. And what would the name of this bacteria be? This would be a streptobacillus, or long chains of those rod-shaped bacteria. Because prokaryotes, and in particular we're talking about bacteria here, but don't forget archaea are also considered prokaryotes. They are everywhere. And again, I mentioned that they do live on and in our bodies and other organisms' bodies. And they play a very essential function for us and other plants, animals, and fungi. They are called the microbiome. So, of course, micro for tiny, biome for life. So this is the tiny version of ourselves, if you will. So the microbiome is the community of microbes that live on and in our bodies. So be able to recognize that term for sure, be able to define it. The intestinal microbiome help us digest food and extract nutrients. The microbiome of our skin decompose dead cells and also act as a line of defense for pathogens. They also can aid our immune system by helping to guard the body against bad types of microbes like viruses or pathogenic bacteria like that Streptococcus aureus or that staph that we learned about earlier. They are also very important in our food industry. So Acteobacter is a type of bacteria that helps ferment chocolate, hops to give us beer, even certain types of coffees and liquor. Arthrobacter helps in the cheese making process. Aspergillus is actually a type of fungi, but other bacteria can aid in the alcohol production. Lactobacillus or lactobacilli is found on and in fruits, which is extremely important bacteria for our own digestive system, and our own pH balance in our bodies. Then we have all types of bacteria in the dairy making process to help ferment that dairy to give us yogurts, milks, other types of cheeses. And then we have bacteria like Enterococci, Halomonas, etc. that aid in like meat preservation, fish preservation, deli meat type foods, okay, all utilize prokaryotes and in particular bacteria. Even in the environment, these organisms are essential to all life. Without these organisms, life could not function or even exist at this point. So there are microbes living in the soil. So every time you go outside and you're walking on soil, that is life in that soil. The microbes living in the soil and at the bottoms of bodies of water help to decompose dead organisms to recycle nutrients. And without this essential nutrient cycling, plants could not live, and then in turn, animals could not live because animals eat the plants. So, extremely important functions in the microbiome, the food industry, and of course the environment in terms of nutrient cycling. So, definitely be able to tell me about those three ecological functions of our prokaryote friends and their application to why they're so important. So those were examples of good type of bacteria, good type of prokaryotes that help us, that we need, that are essential. But usually, especially post-COVID era, if you were to say bacteria, that usually brings up a negative connotation, right? Because well, bacteria can and do make us sick. There are good, neutral, and bad. So we're gonna lean into some of the bad ones now. Some bacteria have the ability to form a biofilm, and this is where they aggregate into colonies and attach to surfaces. Biofilms can form on anything, and they are super tough. They can form on rocks, organic material, even metal and plastic, and even our own bodies. So the plaque on our teeth is a biofilm. So of course this is why do you brush your teeth, right? To remove the plaque. Because what happens if you don't do that? That plaque will form a very hard biofilm on your teeth, and that will harden and calcify into what we call tartar. That's what the dentist will scrape off of your teeth in a cleaning. And that tartar will solidify on your teeth and eventually form a cavity. So those microbes will just eat into the enamel, eat into the tooth material, because it forms this almost impenetrable biofilm that can only be removed by physically scraping it off. If you've ever had a UTI or an ear infection, that is also an infection of a biofilm forming bacteria. So why do UTIs, why ear infections, you have to go to the doctor for those? Because they're very hard to get rid of. The body has a hard time getting rid of it on its own because those bacteria literally adhere to the surface of your bodies with this biofilm. And the biofilm also helps to protect these bacteria against our immune system. So that's why you typically have to go get a strong antibiotic to clear that. Our own bodies have a hard time doing that themselves because it's a biofilm there. How prokaryotes reproduce. So think back to mitosis and that's it. Okay, they can reproduce sexually, and they do often, but the easier route and the more prolific way that they reproduce is called binary fission. And that's literally just mitosis, but we can't call it mitosis because mitosis, we dealt with a nuclear division and we don't have a nucleus here, so we just call it binary fission, which literally means two separations. Okay, so everything in the cell will double and then the cell will cleave into two, and then there you go. Identical, similar to cloning, although little mistakes can accumulate, etc. So the cells continuously copy the DNA, they divide continuously. This is a asexual process. Again, each time this happens, though, there are small mistakes in the DNA copying machinery, and that can lead to random mutation. And if that random mutation is beneficial, then you've led to a new type of adaptation for that type of bacteria. And because these organisms are so tiny and they can reproduce this way so quickly, a lot of genetic variation can occur. So this is one way that antibiotic resistance develops, because these bacteria, because they divide so fast and so frequently, one of those can develop an immunity to an antibiotic. And then that antibiotic will kill off the rest, and then that new resistant strain will keep dividing, and that's how you develop antibiotic resistance. Just like in that MRSA example I mentioned, that Staph aureus divided and developed a mutation to make it immune to that methicillin antibiotic. This will continue very rapidly until resources run out. So either space is limited or they've used up all the nutrients, etc. But as long as everything's in favorable condition, these guys can reproduce almost indefinitely over and over and over again with really nothing stopping it. So they are really masters of reproduction. Some prokaryotes are able to survive extremely harsh conditions. These type of bacteria are able to form specialized cells that we call endospores. So endo, of course, for inner, and then spore is basically an impenetrable, thickly coated cell. So the technical definition, endospore is a thick-coated protective cell produced within a prokaryote when conditions become unfavorable. The endospore can survive trauma and extremes. So if something happens to an endospore bacteria, let's say some Lysol were sprayed on it, and that bacteria detected, oh crap, I'm about to die, it will immediately form a protective coating around its DNA to save that DNA from getting destroyed. When the conditions become favorable again, okay, that endospore will dissolve and the cell will regenerate, which makes endospore forming bacteria extremely notorious and highly pathogenic. They're very dangerous because of this. They're hard to kill. One of the only ways you can kill an endospore is called autoclaving, which is basically incineration in a very special oven that gets to extremely hot temperatures. Those are usually found in microbiology labs, though. Some examples of the endospore forming bacteria. Notice that a lot of the endospore forming species are types of bacilli. So they're the Bacillus genera. We have Bacillus anthracis, which is what will give you anthrax. So back when 9/11 happened, it was 2001. Right after that, the nation was on high alert because, we were scared of terrorists. The government told the people that terrorists were lacing American mail with anthrax, which was like a white powder that was the endospore of the Bacillus anthracis bacteria. And if you opened a piece of mail with that anthracis bacteria endospore, it would essentially create big sores and could get into your bloodstream and kill you. Okay, so that was the news of the day that had everyone on high alert back then. That anthrax, and that was just this bacteria or that endospore forming species. Clostridium botulinum gives you botulism, so it produces a toxin and can form an endospore, so it's doubly dangerous. And also Clostridium tetani, the bacteria that can give you tetanus. Again, has a very toxic, neurotoxic poison that essentially paralyzes you, and it forms an endospore. So all three of these are just examples of bacteria that are highly toxic and they're very hard to kill, which is what makes them extremely dangerous. Ecological impact. So those were some examples of some negative connotations, okay, when it comes to bacteria. But again, another really beneficial service that these guys can give us is bioremediation. So this is another service that they do for us. So again, just to recap, they could cycle chemicals in the soil, they decompose, and we can also use them to clean up our own messes. So back in 2010, if you recall, if you were born at that point or if you were a kid or teenager, BP, which is the big oil company that drills in the Gulf of Mexico, they had a huge oil spill. So one of their oil tankers or oil drills broke, I think, and just dumped hundreds of thousands of gallons of crude oil into the Gulf of Mexico, which was a environmental disaster for one. Killed a lot of the life, destroyed the water. It was a mess. And the one of the only ways to actually clean up that mess that humans caused was to use a microbe or bacteria that ate the oil. And so using microbes to help restore the environment in some way is called bioremediation. So of course, bio for life, remediation is to fix or to put back together. A little bit about the classification here. So again, we've looked at a phylogenetic tree like this in the last lecture, but our prokaryote friends are 2 out of the 3 domains of life. So again, there are a lot of prokaryotes out there. Most of life is a prokaryote in some form, so 2 out of 3 domains. Domain bacteria and domain archaea. And then, of course, this very first the trunk of a phylogenetic tree denotes that last universal common ancestor. Then we have branch points showing the organismal divergence or where things diverge from one another. And now a little bit about that cryptic domain that you don't hear much about. We are just really beginning to scratch the surface at discovering what they are, their implications, etc. Domain archaea. These are known as extremophiles. So archaea, just like their prokaryote friends bacteria, they are also extremely abundant in a lot of places. But not only are they found everywhere that life is, they are also found in places other life cannot exist. So these are the life forms that inhabit even the most extreme environments on Earth. So that's why they're called extremophiles, literally meaning extreme lovers. These extremophiles can be thermophiles, meaning archaea that love very hot temperatures. Like you will find archaea in volcanic rims and boiling sulfur springs, for example. You have extreme halophiles, which are salt lovers. So Death Valley has a lot of salt in the soil, which is why nothing grows there, but archaea are there. The Dead Sea is so salty that nothing lives except archaea. Okay, and then you also have methanogens, which are archaea that can live in oxygen-free environments with a lot of methane that would usually kill other things. So the bottom of lakes, the bottom of dumps, and even in our own gut. So methanogens are extremely important because those are the ones that really facilitate decomposing dead and decaying or just trash to get the nutrients unlocked from them and then to recycle them back into the environment. So the methanogens are extremely important to nutrient cycling because they can get into places nothing else can. And then some bacteria that cause disease. So we looked at some of the endospore forming species that can definitely cause anthrax, tetanus, botulism, etc. But keep in mind that most bacteria are good for us or they're neutral. They don't help or hurt us. They just exist. But those bacteria that make us sick, we call pathogens. So these are the disease-causing organisms. And the way that bacteria can make you sick is if that bacteria is poisonous, basically. Every time you get sick from a bacterial infection, it's because you're being poisoned by these guys. So there are 2 modes of bacterial poison. Either exotoxins, this is where the bacterial cell will secrete the poison out into the environment, or an endotoxin. This is where the poison is just a part of that bacterial capsule that we learned about earlier. So the most effective way to prevent bacterial disease is sanitation. That's why modern day The prevalence of death from bacterial pathogen has greatly decreased because as civilization has advanced, sanitation has as well. So, sewer systems, obviously, education like washing your hands after you go to the bathroom, and also the rise in antibiotic use has all vastly decreased the disease and the illness that people used to get a lot from bacteria. And even some bacteria are used as biological weapons. So, again, I mentioned that anthrax caused by the Bacillus anthracis was thought to have been used after the 9/11 um, tragedy. And Clostridium botulinum makes the deadliest poison on earth. So, that's a very powerful neurotoxin, which essentially paralyzes your nervous system so that you can't move and eventually can't breathe. But interestingly enough, okay, scientists have been able to extract that toxin from Clostridium botulinum and use it to make wrinkles go away. So if you ever hear of Botox or choose to get it in the future, you are literally injecting that Clostridium botulinum toxin into your muscles of your face because it's a neurotoxin. So what it will do, it will completely relax those muscles, so it'll stop the firing of the nerves. And when the muscles relax, the wrinkles will go away. So you're essentially paralyzing the muscles in your face and when you use Botox, because that's derived from the C. botulinum toxin. Here's a very interesting article about botulism if you would like to read it. It's a CNN article, a little bit going into the science of the C. botulinum bacteria and the toxin that it produces. And then just make sure that you read, you definitely study about the human microbiota in your textbook. That will definitely come up on future quizzes and exams.